let's start. I will ask you all to put your microphone in mute during this period. Then for question and answers, so you will see if it will be nice to have the microphone on. Okay. So this is the first day of the, let's say, first edition. I think we can call like that, of TraCyTalks. As you know, Tracy is for translational cytometry and is the scientific platform for flow cytometry at I3S. And the idea of this TraCyTalks came out in a meeting with Alexandre Salvador that is here, and you can see him. He has the camera on, and and I always need to thank him for these ideas, because we were discussing so to have like a webinar, because nowadays needs to be everything online, about topics that we think that go in line with questions that I've been facing at I3S. Of course, there are a lot of questions and a lot of thoughts. For this time, we decided to talk a little bit about bits. So this will be the theme for now. So, as I was telling you, Alexandre is always a big help in all of this, and is, let's say, co-hosting me as well. And I will ask him to check your questions. So if you have questions during the presentations, please put it in the chat box, and then we try to address them. We'll see if it will be in the end of each presentation, or maybe in the end of the two presentations of today. That will be me and Raquel Maceiras. And then we will be together, me, Raquel, and Alexandre, and try to address all the questions that you have. Okay. And if you already have some questions, you can already address. So, I want to, sorry. I will start this webinar with a presentation about control and Monday morning. So, control and standardization beats for flow cytometry experiments. And I will talk a little bit about beats that I consider that could be useful in all of your experiments, okay? So, and because we need to ask some things before we start, okay? So first, and most of the times you are relying on the members of the flow cytometry facility. You can ask, is my instrument ready to use? Then what data can I generate to have robust results? And because we always have an end, and most of the times the end is to publish, we can already think what data can impress the reviewers, okay? So sometimes we can put that ahead, and it's not to impress, it's also to have accurate results. So the three beats that I think you can always have in your experiments, or you can consider, it's not a must, but you can consider, the quality control beats, counting beats, and compensation beats, okay? So beats are the most common materials used to calibrate, control, and standardize instruments. They are well known. We know the size, we know the fluorescence, so it's the reason that we use them. And quality control is an essential component for quality reproducible data, and should not be just considered for the members of the staff, for us, so me and Emilia, that we are working in the facility, and we do this quality control regularly. But you also can consider them for your own experiments. Okay, you can do this quality control before you start your experiments. So the quality control bits are essential for determining instrument performance, and this is going to tell you how the system is working and making sure that it's working day consistently. But it only makes sense if you track it over time. Let me just, if you track it over time. If you don't track it over time, you won't see the changings, and actually you are not doing quality control. So it's only a quality control if you do it over time. For that, you can use an original equipment manufacturer's protocol, or you can design your own protocol. And you need to see, and so you determine what is important to track. Okay, if you are using a protocol from the manufacturer or your own, you determine what you want to track. So here I start showing you an example of a control program, a quality control program, and this one is from BD. And as you know, all the cytometers that we have in the facility are from BD. And we, in some of them, we run these CST bits, these cytometer setup and tracking bits. And they measure a lot of parameters that are really important to be sure that the instrument is working properly. So it does the laser alignment, it measures the electronical noise, the detector efficiency, the channel background, this is the sensitivity of the detector, and it adjusts the photomultiplier, so the PMT voltages, to maximize the population resolution in each detector. Okay, here I have a really small video, and I apologize already for this video because it's shaking, and also then I put it in a fast forward way, but it's for you to see what it looks like when we run this bit. So we can see here the lasers. In this case, these bits are passing in the sorter, in the FACS-32, and we have these 3 lasers. And while they are running and they are measuring all those parameters, we can see that the bits have different sizes. And then we'll see that they have a different intensities, and they are calculating all those parameters, and in the end, it will tell me if they pass or failed. And sometimes they pass with warnings, and we need to be aware of that. And I will receive a report, and I can track what is happening and spot the trends and see if everything is okay. So here everything was okay. I received a table, and I also can see these graphs. We will talk a little bit more about this because I'm sorry for the video, but it's not the best. So when we receive the equipment, or every time that there is a physical configuration or the cytometer at a periodic maintenance, or for example, that was a change in the bead slot, and that is important. I will say this. So every time that you change your bead slot, you can have alterations. So something that can alter the performance or increase the electronical noise, we do a baseline. So it does all these measurements. To do the baseline, it takes a little bit longer, and we don't do so routinely. But then we do routinely the performance check that basically he measures the variations that can occur for the baseline, okay, comparing with the baseline. Here is an example for 2 detectors. And so the baseline and the performance check are automatically created and contained all the performance measurements determining the CST process, and they display the report, and we can check all, I'm sorry, you can check all the measurements that that was done. For example, the MFI, the CV, the variations in PMT, and the laser alignment as well, and delays. So everything you can check, track, and see that are you can track the trends if they are, if it's something that you should be concerned, if you should keep an eye on that. And also here you can see that we have actually 2 baselines. The blue one is from the previous beat slot, and then the black is from the current beat slot. And sometimes there are some measurements that goes a little bit too out of the baseline, and there is a cross here, a red cross. So we can track this pretty easy with these CST bits. Another example, and actually this one, it's the way we do quality control in Accuracy 6. So here it's your own protocol, and there is a template, and we should have some informations in the template. These peaks here are the rainbow. Actually, these ones is the 8 peaks. So we did a gate on beats, then we see the 8 peaks for, in this case, for these 3 detectors, FL1, 2, and 3. And then we should always have a table where we can follow what is important to be sure that the equipment is working properly and they are within the values that we want to know that is working in proper conditions and everything is going well. So, okay. Quality control, in this case, are the bits. Quality control is critical for ensuring the consistency and the reproducibility of data. Helps to identify issues before they become a problem. Okay, they are only useful if you monitor on a regular basis and ensures the instrument linearity by means of voltage optimization and verifying the fluorescence in each channel. Okay, so the other bits that I mentioned, it is the, I'm sorry, this is, okay, the counting bits, and they can be really useful for your experiments. And let me just, they can be really useful for your experiments, and because most of the times what happens, so you are in your lab and you have a lot of cells, and of course you can take a little bit of your sample and dye with the trypan blue. You put trypan blue and you count in your new power chamber, and it's okay. Of course there always you can always have the human error, but it is what it is. Other thing is some flow cytometers, such as the Accuracy 6, they allow you to do volumetric measurements, and like that you can do the counting and direct calculations of the number and concentration of a sample. But there is also this option where you can add an internal microsphere or these counting beads that they are standard, and you add them to your sample, and then you can count and direct calculate of the number of cells and concentration of your sample. Okay, so usually these beads are sold with a defined concentration, and with this you can do a stop gate, and you and while you are acquiring, so it will stop in the number that you set, and then you can calculate back to your cell concentration in the original sample. So here is the formula that you can use to calculate your the concentration of your sample. It's quite easy, and of course it will be, the pipetting will be crucial. Okay, so okay, here are a table with some beads that you can find in the market. A lot of brands already have. Here are just few, but it's for you to have an idea. Most of them are fluorescent, and they are excited by almost all the lasers that you can find in your flow cytometers, and then you can easily see them and do a gate. Sometimes it's not so easy to see when you put fourth side scatter, but when you when you put against a fluorescence detector, you should be able to see it. Okay, now I want, before I talk a little bit about compensation beads, I want to talk what is spillover and the compensation that are related. So the fluorescence spillover is the amount of signal that the fluorochrome emits in a secondary detector specific for a different fluorochrome. One of the examples is this one here. So when FITC enters in the PE channel when excited by the blue laser, I think a lot of you already faced this. And sometimes this is the headache of the researchers. But we can minimize this spillover. So, and by the compensation is the mathematical process used in all flow cytometers and softwares in which the spillover values are used to determine a compensation matrix that will correct the background due to spillover in all detectors. And as I mentioned, it's a mathematical process. So it's the reason that I will also that that automatic compensation should work really well. So these compensation beads can help you in your experiments. You will save, first you can save cells because you don't need to do your monolabels in cells. You can do it with these beads, and these beads bind antibodies of multiple isotypes for multiple species, allows to use the same fluorochrome used in your experiment. There is no biological material is required, provides sufficiently and accurate spillover values, and is easy to prepare and use. Okay, but there are some considerations that I want you to have in mind. First, the fluorescence spectrum of the compensation control fluorochrome conjugated reagent should be identical to the reagent used in the experiment. Because I have here two examples. Don't use in your monolabel FITC and then in your mix you have GFP or APC and Alexa 647. They will be detected in the same detector, but they don't have the same spectrum. So the best is always to use the same antibody with the same fluorochrome. Other thing is the positive population should be as bright as possible. Okay, if you have a true spillover value, you will have, so the compensation matrix will be correct if it's derived from a bright or a dim positive population. If the brightness is compensated, the dim should be always also compensated, okay? And another thing is the autofluorescence of the positive and the negative populations must be equivalent. A lot of time happens that you bring the unstained cells and use that unstained cells for as a negative for all the controls, and then you are using beads, for example. And the autofluorescence of the negative in beads and in cells can be different. So what I recommend you, and for example, here is the example from Faxtiva. When you are in Faxtiva, you put to do automatic compensation, and then you go there and they design, they draw already a gate for the positive, and then you can adjust. But what I recommend you is also to draw a negative gate, okay? A gate in the negative population. Other consideration, and this consideration I will say for everything actually, it's collect enough events. In this case, to obtain meaningful, accurate SUVs. Okay, so it's always important, because sometimes you are not saving money because you are recording less, because you are using less bits. I will say that is much more expensive if you need to repeat an experiment. And so another thing, so here is a table of available compensation bits that you can find. I'll say try to, because there are now different bits and sometimes not all of them will fit in your experiment. And you can already, so you have here some examples. These ones are very bright. And if you enter and you see the data sheet of them, you can already see why they did this. So try to study a little bit what bits will fit better in your experiment. And then try to test them, and don't change in the middle. For example, now you are using one from one company and then you change for another, and the experiment will be the same. So you are repeating the same kind of experiment. So do not do this kind of changings that can compromise your experiments, and then you will see differences between them. Other thing is you are using from the same company the bits, but for example, in that day you need to do 6 controls so 6 tubes with these bits, but you only have for 4. So you have another vial in the fridge, and you do 4 with the old one and 2 with the new. I will say that you should use in that day all from the same lot. So maybe it's better to use the bits from the new vial for all, instead of using half and half or for 2. So be careful with that, because sometimes you have changes and you can avoid that. So just to finish, and this is for all bits. So bits are small particles with well-defined size and fluorescence, which can be used for PMT voltage optimization, compensation, setup, cell counting, scale calibration, and so on. And bit controls and standard helps ensure that your data instrument is operating properly and that your data is both reproducible and reliable. And just to finish, I want to thank you and to tell you that of course you always can contact me, and I also put here Emilia, that she's always a big help in big help. She's a member. She's so I need to always to thank her, and thank you for registering, participating in this webinar. Okay, I don't know if there is already questions that I should address now, or if we...
No. Until now, no questions.
Till now, no questions. Okay, it's no doubts, I believe so.
So regarding these questions, I want to make just one important comment.
Yeah, please.
In the publications that, for example, most of you want to do right now, all Nature group has accepted what we call the MyFlowSite protocols. This means that they will ask you each time that you want, for example, to publish some flow cytometry data. They will ask you exactly which kind of instrument, what kind of information do you have, how did you do your gating strategy, the controls, and so on. So this information you can collect from the unit. The Tracy will provide you all this information, but also if they have any doubt on this, it will be very important for the persons to understand in those days that they made the experiment, if exactly the instruments, how they were, how did they perform. So you can go back and you can actually see, because all this data that Catarina was showing is registered in the instrument. So you can go back and you can actually understand how it's done and the feedback that you have. So this information is also available, okay, and you can have a look at it. I don't know, I don't think I can share my screen, but if someone...
You can.
Okay.
Me. Now you can.
Okay, let me just share my screen to show you exactly this. So you can find this MyFlowSite, okay? It's the minimum information that you can provide in a flow cytometry experiment, okay? All this information, and what you can have in the end is a simple document like this, very easy to fill, okay? It will be very easy. It's accepted by the ISAC community, nature reviewers, and so on. And actually, in the end, the only thing that you need to do is, for example, like this is just an example, okay? You tell the purpose of your experiment, the keywords, all the variables that you have, the organization, contacts, all this. But then you start putting here some of the information, saying what kind of instrument that you did, the detailed protocol, and then instrument related details. Who was the manufacturer? Which model? The configuration, because you know, different instruments, different lasers, the optical configuration and filters that you used, which fluorochromes did you use? So this kind of information will be, let's say, in the supplementary information. Right now, it's, I think, more important than the article by itself if you want to reproduce the data. So it will be very important for you to be sure that the instrument that you have used is in the proper conditions. This is the main point. So you can have confidence in the data that you are using. So I think that part it's the crucial part. Okay.
Thank you. It's I think it's a really useful information. So since there are no questions, I will pass for Raquel Maceiras, that will talk a little bit how she uses this kind of bits in her own experiments. Raquel Maceiras is now a junior researcher in the immune regulation group, and she's been working with flow cytometry for several years, not only at I3S, but before at IMM during her PhD. So I want to thank her for accepting this invitation, and go and that you can start. Thank you so much, Raquel.
Okay, first of all, I want to thank the invitation for being here. I will talk a bit about counting and compensation beats, at least from my knowledge and from what I've been using. Of course, there may be other approaches and other uses, but I will talk a bit of what I've been doing. I will repeat a bit of Catarina said, but please bear with me. So counting beats are important to get absolute counts of the cells that you are acquiring or other samples that you are acquiring on a flow cytometer. Actually, when I started, the beats that I used were not fluorescent, so I had to adjust to 4 and side scatter in order to get them. But nowadays, most counting beats are fluorescent, so you can actually disregard them when you're selecting your 4 and side scatter voltages, because then you will find them because they will be fluorescence in most channels, as you can see here, and across all most of the lasers. So the counts, for me, the easiest way to see it is that you want to know how many cells you have in your tube, and you can calculate that based on the cell events that you acquire and your bead events. And that ratio multiplied by the amount of beads that you added to your tube, you will get your total cell count. Of course, the bead counts will depend on the beads concentration that is determined by the manufacturer and the volume of beads that you use that can be adjusted. Of course, if you have many cells, you will have to use more beads. Otherwise, you will have very few events. But if you have very few cells, you can adjust the beads volume accordingly. Okay, so what are the uses that the applications that I've been using? So I've used them on blood sample analysis, where I want to understand the amount of cells that I have on a specific volume of blood, in this case of mouse blood, but this can also be used, for example, for human blood samples. This is also very nice when you have a very limited or very few cells. For example, after cell culture, you want to know whether your cells, for example, proliferated or not, and you want to know how many cells you had at the end of culture. And if you count them, you will not have cells to do your FACS analysis. So in this way, you can have both results simultaneously. Another application that I've used is after cell population enrichment, when you have a mixture of cells and you enrich for some of them. For example, when you're analyzing progenitors or stem cells within a population, so you just remove all the non-progenitors or non-stem cells, but even so you have a mixture and you want to analyze it by flow cytometry and you want to know how many cells you have of each type, you can also use these counting beats. And of course, if you have a very large number of samples and you have a limit time, it will be much easier than count by hand on a Neubauer chamber 30, 60 samples. I don't know how many you can have in one experiment. So this will in this way, it will be easier. Of course, you will have to have an idea at least of how many cells and how many cells you're, for example, staining. But for have a precise count, it will make your life easier, that's for sure. I just wanted to make this note because you're counting the amount of cells that you have at acquisition in your tool. So if you have a lot of steps of washing and staining and fixing cells and so on, there are some protocols that advise you to add the beads prior to that, so that the beads also go through all those rounds of your experiment. It really depends on how exact you want your counts, because if you want to know more or less how many cells of each type you want to know, adding the beads prior to acquisition should be more than enough. Okay, so regarding compensation beads. So how do they work? So these beads have antibodies on their surface that will bind the antibodies that you will use to stain your samples. However, I think it's important to note that they will bind the non-variable part of the antibodies. Okay, so these not depend on the specificity of the antibodies that you are using. So in this case, you have to adjust the site for the site scatter during compensation. But since these are the two parameters that you can change later on without affecting compensation, you can always adjust it later. And of course, you get a positive peak from the beads that bound the antibodies, and a negative peak from control beads that do not bind the antibodies. Okay, so when do I use compensation beads? When I have a limited amount of cells. So if I don't have cells to do my experiment and to do the single stainings, why not use compensation beads? Another important way is when you use, for example, you want to sort fluorescent cells or cells isolated from a reporter mouse. So these cells are already fluorescent, so you cannot use them for compensation. In this case, you can use compensation beads. It can also be used if you're not sure if your cells express a certain marker, because you know that you will have a positive peak. However, I put there the exclamation mark because I want to know that, of course, you'll have to have positive controls. Otherwise, you will not know if it was your experiment that failed, or what is the reason why you don't have the staining working, or a positive in your samples. For example, if you have a very rare population that express the marker, so imagine that you're using an antibody that only binds to 0.01% of your cells. So the amount of events that you have to acquire in order to have a peak to be able to do the compensations would be a very large amount of events. So using beads, you will not have that kind of problem. This is, for example, right now in the lab, we are working at IBCL 3, so in biosafety level 3 facility, and it is mandatory that you reduce the amount of work that you perform under those conditions. So for us, it's much more easier, especially when you have a very large experiment and you're performing a lot of stainings with many colors. It's just easier to make the compensation, the single color for compensations outside the biosafety level 3. And you can do it with the same reagents, but in the lab, so that you don't spend so many hours working at CAT3 facility. And also, as you can see here, these are very nice and quite nice peaks that facilitate the compensation. If you have a smear of a population, or if the peak is not very well defined, it's more difficult to compensate. So using beads usually it's easier to perform the compensation. Okay, regarding problems, this is just one example, is to use the right beads in your experiment. For example, these beads that I've used, they are not quite good. For example, for this channel, this is Pacific Blue or Brilliant Violet 421, what happens is that the beads are quite fluorescent for this channel. And so it's much difficult to adjust the right voltages so that when you go to your samples, you also can discriminate the positive and the negative in your samples. So of course, if you're using secondary antibodies, only these ones are used to stain the compensation beads, because these ones are the ones that are fluorescent and will be and can bind the beads so that you get the signal. Regarding other problems, don't assume that just because you have 2 peaks, they will work perfectly on the samples, because one antibody can work fine because it is recognized by the beads, but then it may not bind effectively your samples, because that depends on the specificity, and it will also depend on how much of the antigen you have on that on your sample. So how much surface marker or whatever it is that you're staining is expressed on your cell. So the 2 peaks being very nice when using compensation beads doesn't mean that will work very well on your samples. And on the same idea, you cannot test and titrate an antibody with beads, again, for the same reasons. The beads will depend on the binding to the non-variable region of the antibodies, while the antibodies will depend on their specificity to the marker that you want to stain. You cannot use beads to compensate cell proliferation dyes. For that, you need probably DNA. So you'll need cells. And in the case of fixable viability dyes, there is already beads that can be used for these, but they are not the same beads that you use for antibodies. So in our case, we still use cells when compensating fixable viability dyes. And of course, they work with the almost any antibodies, but there are some antibodies from some hosts that may not be recognized by these beads. And that's all that I had to show you today. I hope I explain a bit of what I've been doing and how you can use both of these beads in your experiments at ITS. Thank you.
Thank you, Raquel. And actually, I think you had some points that I didn't mention, and I think it's good. And not every time compensation beads are the best for single stainings, it's true. And I want to mention that when you are starting and you are still seeing what will be the best for your experiments and the best controls, sometimes you cannot use, so the beads will never be the best, and you need to use cells, for instance, for your positive control for your single staining. So the best is always to test, to do those studies. So first is to do all the controls with cells, and in this case for singles also with beads, and then see what fits the best, then to proceed for the other experiments. I will say that. No, I'm seeing there is no questions. So this last part of question, you have?
But let me, no, just some comments.
I hope I didn't say anything wrong.
No, I just wanted to highlight 2 things, and I think it's important. First of all, is one of the things that you said, it was very important. For example, it's very useful to use this kind of compensation beads. For example, with unknown markers. For example, when you don't know the level of expression. And especially, for example, in activation markers. When you go through an experiment, when you need to activate cells, usually people don't have good controls. So usually they need to activate the cells. So since day 1, they don't have the proper controls. So one of the things that you can overcome is this difficulty using the compensation beads. And to get also to the mind of the people, saying that you can actually mix in an experiment some of the compensation controls with beads and others with cells. So you don't need to do everything in beads or everything with cells, so you can mix them. And related to that, there is one comment that you mentioned, for example, for the channel of the 421, for example, the violet 421. Sometimes the beads, for example, because of the instrument settings that you have, because you adjust the negative with your negative sample, and the voltage are defined, so you don't change voltage anymore. So after that, sometimes you pass the compensation beads and you see that the negative has moved. So it's not like you wanted, but it's there. So one of the things that you can do is actually tell, because it's like Catarina said, a mathematic issue, then you say where now your negative is. So there is a possibility, don't delete regions, but actually you can put a new cursor saying that you have a new population called P3. So then you go and you add a P3 population to the negative one. So mathematically, what he will know is that now he will need to take into account that in that tube, your negative has a new median. Okay, so it will calculate properly. If you don't do this, sometimes what you have is an overcompensation. And sometimes people come to us and say, oh, the beads didn't work and the compensation is very bad because they don't adjust this negative population. So you need to adjust this new negative, let's say, because it's beads. It's not like the cells that you have. Once you have cells, compare with cells, and then you compare beads with beads. That's the proper thing. So when you compare beads, now your negative bead is in another position than your cells. So you can compare cells with cells, beads with beads. So that's the only comment that I have. And actually it will work. One other thing that it's important is for the persons, you mentioned there one thing that it's very important. Once you start an experiment with a compensation bead, okay, don't think that all beads from all vendors are the same. Okay, so if you start with a company, stick with that company because it will be important to have reproducible results. So you need to reproduce. And Catarina mentioned two kinds of, for example, compensation beads. From BD, for example, the normal compensation beads and the plus. Okay, the plus are used, for example, when you have a very autofluorescence type of cell, or for example, cells in culture. So because it has higher levels of fluorescence. Okay, so it's important to choose the beads according to the type of cells that you're using and the kind of experiment. So it will fit more properly the experiment that you're having. So I think that's important also to emphasize to the people. So I don't know if there are any questions regarding this kind of usefulness of the beads.
Yes, that will be. Actually, when I talk about change in the end, then I was running to time of time, and but it is exactly that. So stick to that company when we think that is the best. Don't, if you have different change lots, don't change it in the middle of an experiment. So try that your experiments are comparably longitudinal. In a longitudinal way, I think it's a good message. But any comments? So there is nothing in the chat. Any comments? Any problems that you've been facing? Something that you want to start? I don't think. You can also use the microphone if you want instead of writing. No, nothing.
That's a clear message.
Yeah, are you sure that was clear? We won't repeat this. Okay, so I don't know if do you want to make a picture of today? Because we will have, I didn't mention that in the beginning, we will have 2 more days, as you know. So the next 2 Mondays, the next one, so on the 15th of March, will be Alexandre. He will be here.
And Monia.
And Monia, I will never forget Monia. They will talk about immune-based immunoassays, and then we'll have also Salome Pinho and Inês Alves showing how they use them in our own experiments. I think will be, so we talk already a little bit about capture beats, in this case the compensation beats, and then we'll proceed with those capture beats. And I hope that will be, and I hope that will be more question and answers for the discussion.
They can send the questions prior to the presentations if they want. And also we'll have Andre Mos and Ricardo Jara after. So they will talk also about some other applications with beads that you can actually have. So I think that one of the critical parts in here is beads can help you in setting up the instrument, making it reproducible. Andrea is also a fan of beads. And for example, for the Euroflow project and consortium, for you to have an idea, all the instruments around Portugal and other countries. Okay, so you can compare results. I've also done the setup of the instruments with beads, so you can have reproducible results across all the world. So everybody that uses Euroflow consortium protocol need to use the SOPs, and starting with the setup of the instrument. First, they run the CST beads and check if the instrument is okay. Then they start by calibrating the instrument with beads with a known lot, so it can compare with target values. So you can do that because, for example, this setup is crucial in a long time experiment. For example, imagining that you are following your patients or your mouse during 2 or 3 years, for example, during all the PhD, it's important to have a reproducible instrument. So that part is crucial. So beads can also be very useful for you to target the instrument and monitor the settings, because instruments fail. So the laser is changed, the PMTs are realigned, and then you need to reproduce the data. And that part is crucial, for example, for the instrument. So it's one big usage of the beads. So I encourage you also to control and monitor your settings, okay? And for that, the only way you can do it is using beads.
Yeah, that can be the CSTs. So CSTs can be used not only for the instrument, the first part, but also on that part, or rainbow beads, or there are a lot of beads in the market.
So we take the picture. Everybody puts the camera on and we take the picture.
I will check if I know how to take a picture of this. Let me see. I need to change the visualization of this.
So we have a foreign guy that it's almost one of the only ones that is using a flow cytometer right now. Andre is using the flow cytometer at the moment.
Yes, Andre is with us, Andre Moses, and she's using, she's doing a sorting, but always with an eye on that. I will try into gallery. Okay. I need to take 2 pictures, and I'm trying to see where I can take a picture. Alshan?
Screen.
Okay, that I know. That is pretty easy. Okay.